Hello everyone. Welcome to today's webinar. I'm Jeff Huber, a contributing editor with CNN Media Group, and I will be moderating today's event. This webinar is titled Oligonucleotide Analysis Simplified, MALDI and ESI Solutions, and is being sponsored by Bruker. CNN works with sponsors to identify topics of interest and value to CNN's audience, and consistent with CNN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of the screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You are encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars, and that each webinar will be archived at CNN Online after the live webcast. The presentation today is being sponsored by Bruker. Bruker strives to be a performance leader in life sciences and analytical systems. For over 50 years, Bruker has been driven by a single idea: to provide the best technological solution for each analytical task. Its trusted solutions encompass a wide number of analytical techniques, ranging from magnetic resonance to mass spectrometry and gas chromatography to microanalysis, optical, and X-ray spectroscopy. These market and technology-leading products are driving and facilitating many key application areas, such as life sciences research, pharmaceutical analysis, applied analytical chemistry applications, materials research, and nanotechnology, clinical research, molecular diagnostics, and homeland defense. During the presentation, we will hear from Anjali Alving. An analytical scientist by training, Anjali has been working at Bruker Deltonics for more than 9 years. She has broad experience with bioanalytical mass spectrometry, focusing mainly on Bruker's ESI, UHR, QTOFs, and ion trap instruments. In her current capacity as a senior scientist in the BioPharma Group, her work centers on performing analyses of samples utilizing QTOF and ion mobility mass spectrometry-based workflows for oligonucleotide analysis, characterization of intact proteins, peptide mapping, HCP analysis, and glycan and polymer analysis. She has also authored technical documents to highlight new and innovative application results using Bruker's ESI MS systems. And with that, I will now hand things over to Anjali.
Thank you, Jeff, for the kind introduction, and thank you all for your participation. Today, we will talk about oligonucleotide analysis by mass spectrometry, the challenges that we encounter and solutions that are offered in terms of hardware as well as software. We will also look at examples of oligonucleotide analysis from different companies. I'd like to begin this topic with a rather special story that many of you may have heard. This is Mila's story. In late 2016, Mila, who was then a 6-year-old child, was diagnosed with Batten disease. This is a rare and progressively fatal disorder of neuronal lysosomal storage. The mutation that Mila had inherited was a retrotransposon, which is a 2,000-letter stretch of code that had moved from one portion of her genome into her CLN7 gene and inserted itself there, altering it. This was therefore impeding the function of a critical housekeeping gene in Mila's cell. Dr. Timothy Yu, a neurologist at Boston Children's Hospital, learned about Mila's story and led the development of a novel antisense oligonucleotide drug in record time in less than a year. This story that appeared in CNE News last November highlights the potential and promise of oligonucleotides in personalized medicine. Having thus emphasized the importance of oligonucleotides, let's take a step back now to some very basic information. Oligonucleotides or oligos for short are synthetic strands of DNA or RNA. They are produced through a well-established automated solid phase synthesis process that uses phosphoramidite building blocks derived from deoxynucleosides and ribonucleosides. The DNA double-stranded helix in the figure that you see shows the complementary pairing of adenine to thymine and guanine to cytosine. In RNA, the thymine is replaced by uracil. Oligonucleotides have various applications from research to disease diagnosis and therapeutics. For example, primers used in PCR and DNA sequences are short single-stranded DNA, typically 17 to 25 nucleotides, that match the beginning and the end of the DNA fragment to be amplified. PCR assays and panels are designed to detect and measure the expression of biologically related genes. Fluorescence in situ hybridation, or FISH for short, is used to detect and localize either DNA or RNA within the cells and tissues. Oligonucleotide therapeutics are a novel class of biopharmaceutical products comprised of short strings of synthetic nucleotides designed to alter the expression of target genes associated with disease in the body. Let's take a closer look at this class of applications. Looking at the three different types of oligonucleotides as therapeutics, we see that we have antisense oligonucleotides, also known as ASOs. These are short synthetic single-stranded oligodeoxynucleotides that can alter RNA and thereby reduce, restore, or modify protein expression through several distinct mechanisms. The drug that was approved by the FDA, fomibursin, was developed by Isis Pharmaceuticals, now known as Ionis Pharmaceuticals, and marketed by Novartis. This is an example of an antisense oligonucleotide drug. Next, we have the small interfering RNA, or siRNA. These are double-stranded RNA molecules, about 20 to 25 base pairs in length, that interfere with the translation of proteins by binding to and promoting the degradation of messenger RNA, also known as mRNA, at specific sequences. The example here is patisiran, which was developed by Alnylam Pharmaceuticals. The third category is an aptamer. Aptamers are short, single-stranded DNA or RNA molecules that can bind to a target. This could be a protein, a peptide, carbohydrates, or small molecules. They bind with high specificity and selectivity. The binding is determined by the tertiary structure rather than the primary sequence. And the example here is pegatinib, which was developed by OSI Pharma. Oligonucleotides are produced through an automated solid phase synthesis process that is well established. However, one can encounter various impurities. As seen in this table here, the sequence failures. For a 20-mer, one can expect 10% sequence failure at 99.5% synthesis coupling efficiency. Other impurities include residual protecting groups and byproducts from modified bases. There are several challenges that one comes across in oligonucleotide analysis. For example, there is a huge complexity and diversity of oligonucleotides. These are differences in size, chemical modifications, or conjugation. Another issue could be the interference of the encapsulation or targeting component of the drug. A great deal of effort thus goes into method development of a specific compound. Accurate molecular weight determination and confirmation of the nucleotide sequence of an oligonucleotide are essential in confirmation of the identity of the molecule, and this is therefore a regulatory expectation. Mass spectrometry is the gold standard for oligonucleotide QC and characterization. ESI, or electrospray ionization, and MALDI both have their strengths and limitations for oligonucleotide analysis. The figure on the left shows that MALDI is well suited to high throughput analysis with thousands of samples per day. It is also tolerant of samples with high amounts of salts or additives, and best results are obtained with oligonucleotides less than 50 bases. The figure on the right shows that ESI LC-MS can handle about tens to hundreds of samples per day. Larger oligonucleotides, ranging from 20 to 120 bases long, benefit with the multiply charged envelope obtained in ESI. In addition, since ESI is a relatively soft ionization source, it is better suited to analyze photosensitive or fragile oligonucleotides. Bruker offers a range of different MALDI MS instruments, including high-resolution MALDI TOF and TOF modules. On the left, you can see the Microflex MS system with a resolution of 15,000. Next, we have the AutoFlex Max, which operates both in MS as well as MS mode with 26,000 resolution. The UltraFlex Extreme again operates in MS as well as MS mode and has a resolution of 40,000. The RabiFlex is the top of the line instrument for MALDI, which has the ultimate resolution, speed, and sensitivity. It has a specification of 50,000 for resolution and a 1 ppm mass accuracy. Let's look a bit closer at the AutoFlex Max, which is one of the best suited instruments for oligonucleotide screening due to its scalability from linear mode to reflectron mode to TOF mode. The AutoFlex Max MALDI TOF system has some nice new features, such as a flash detector with a new 10-bit digitizer. This is important for low ppm mass accuracy and about 4 times more dynamic range. The proprietary PAN technology helps in a high resolving power of up to 26,000 with attomole sensitivity. And we also have the SmartBeam 2 proprietary solid-state laser technology, which is the best-in-class ionization for a wide range of MALDI matrices. On the electrospray ionization side, Bruker has several high-resolution QTOF instruments, starting from the compact on the left, which is a benchtop instrument with a specification of 30,000 resolution and 1 ppm mass accuracy. Going up, we have the Impact 2 system with a resolution of 50,000 and sub-ppm mass accuracy. The Maxis 2 ETD MS system is the top of the line with a resolution of 80,000 and 0.6 ppm mass accuracy. The new timsTOF Pro that is based on the Impact model has an added dimension of ion mobility and a superior ion mobility resolution of 200. All of the QTOFs are well suited for oligonucleotide analysis, but typically the Compact or the Impact 2 with the elute analytical flow UPLC system is used. Again, BioPharma Compass software is the software of choice for analyzing both the MALDI as well as the electrospray oligonucleotide data. This slide shows the hardware of an ultra-high resolution QTOF mass spectrometer. Beginning on the left, the red line traces the ion path from the electrospray ionization source through the glass capillary and to the dual ion funnel, followed by the quadrupole mass analyzer, the collision cell where fragmentation takes place, and into the flight tube where the ions are reflected with a single pass reflector down to the detector. The flight tube has in-flight focusing for full sensitivity resolution, and the detector has a 10-bit digitizer that increases the dynamic range. BioPharma Compass software is a wizard-driven platform for automated MS data acquisition and processing. Analyses can be set up via a simple web interface, and methods can be chosen from several ready-to-use workflows. There are different powerful data analysis and reporting tools to process the acquired data and generate customized reports that can be emailed directly to the operator. The software platform is fully supported by a 21 CFR Part 11 compliance-ready framework. BioPharma Compass is also unique in its ability to seamlessly unify MALDI TOF and ESI QTOF data, as well as fluorescence detection and UV data, and transform it into meaningful knowledge. BioPharma Compass comprises several workflows tailored to analyze the specific compounds of interest. The multi-target screening workflow supports the analysis of both electrospray and MALDI data acquired for oligonucleotide and peptide analysis. Other workflows include the protein screening for intact protein or subunit analysis, the top-down or middle-down sequencing for ESI and MALDI, and the peptide mapping analyze digested proteins at a peptide level for PTM analysis. The multi-target screening electrospray ionization workflow is a wizard that guides you through a setup of a new method or allows you to process the data sets with the previously established method. The screenshot below shows the first step, where several data files that have been imported previously are now ready to be processed. This slide shows the sample table. The sample table provides information for each specific sample spot so that the analytical results can be related to a sample unique ID. Also note that the formula or the molecular weight can be provided for all of the compounds. The workflow parameters of interest include modifications, chromatogram and spectra processing parameters, any background masses or adducts, multi-attribute monitoring, as well as peak list processing parameters. The table at the top shows the typical modifications for oligonucleotide analysis, such as depurination and depurimidation on the bases. The lower table shows the typical salt adducts that are seen, such as sodium and potassium. Both of these tables can be customized to include the modification or adduct of interest. Multi-attribute monitoring is an important feature of the workflow. For the oligonucleotide QC workflow, the following attributes that you see below in the table are defined. The base peak as expected means that the base peak as defined in the modification profile must be the base peak that is seen. Target profile matching. In an ideal world, the requested profile and the profile displayed in the spectrum do match. But if chemical modifications such as depurinations come into play, then this is not the case, and the cosine factor or the cosine score provides a hint for this criteria. Mass accuracy is a crucial attribute for the quality of the measurement and calibration. The intensity coverage, which is seen in the last row, is a parameter that defines what fraction of the intensity of all peaks in the peak list should find itself in the modification profile. This parameter allows quantification of unexpected peak abundances, such as those derived by early truncations during synthesis or any unusual contaminations that might be present in the sample. The actual results table view provides access to the full information seen in the sample table as defined in the method wizard. It also shows the multiple attribute monitoring icon for a complete overview of all the sample analyses. The result visualization works with several views that relate to each other. So clicking on one of the rows leads you to a detailed result in the expected view. For each modification profile compound, as defined in the wizard, the matching information is given and a red or green color coding is assigned to each observed compound, whether it was expected in the sample or not. A comparison of the reference and measured profiles, as shown by the two blue arrows, is also possible. This slide illustrates the automated acquisition function. We begin on the left with the tab that allows queuing up a new batch of samples for analysis. You can then enter the information and then queue up the different batches. Once the analysis is completed, the Batch Processing tab displays the status as done. As can be seen on this slide, reports can be customized to include several different parameters, such as the experimental parameters set by the user, chromatograms, MS spectra, and results based on criteria defined by the user. Let's take a look now at the examples provided by some companies that are involved in oligonucleotide analysis. We will begin with Execure. Execure Incorporated is a clinical stage biotechnology company developing a new class of immunomodulatory and gene-regulating drugs against validated targets. They have a proprietary spherical nucleic acid formula. The inflammatory diseases, genetic disorders, and oncology are some of the lead programs for which they develop drugs against. The bulk of the oligonucleotide samples are analyzed for accurate molecular weight verification. There are numerous modifications within the sequences, however, that are specifically monitored based on theoretical mass, followed by determination of the contaminant when this mass is not within the specified limit. For example, depurination, a PO to PS substitution, or protecting groups, all are examples of modifications. These analyses are done on the ultra-high resolution compact mass spectrometer. Let's take a look at analysis of failure sequences. High purity is a requirement in oligonucleotides used as therapeutics, and identification of failure sequences and other impurities is critical. During oligonucleotide synthesis, Even with the most efficient chemistry, it's difficult to achieve 100% reaction efficiency, thus causing some of the unreacted 5' hydroxyl groups on the resin-bound nucleotide. The following resulting oligonucleotide would thus lack one base and would correspond to a deletion mutation of the desired oligonucleotide. As you can see in this figure, the correct sequence is seen on the top row, while the bottom row shows a failure sequence corresponding to the deletion of a thymine residue. The figure on the top shows the molecular ion at 6669 Daltons, and focusing on the highlighted region, which is seen in the bottom figure, we can see the loss of guanine corresponding to -151 Daltons and the loss of cytosine corresponding to -111 Daltons. The next modification is analysis of the phosphorothioate backbone. DNA and RNA are often modified to increase in vivo stability, uptake, and toxicology. One of the common modifications is the incorporation of the phosphorothioate linkages. As seen in this figure, the phosphorothioate bond substitutes a sulfur atom for a non-bridging oxygen in the phosphate backbone of an oligo. The figure on top shows a peak at 7835 Daltons that correlates to a loss of -16 Daltons for a PSPO substitution. And in the lower figure, in addition to the single PSPO substitution, we can also see a peak correlating to -32 Daltons for a 2 times PSPO substitution. This slide shows a figure where a 70 Dalton mass shift is seen for the isobutyryl protecting group. Next, let's look at a major oligonucleotide reagent manufacturer. They use MALDI for their high throughput quality control. This company provides custom DNA and RNA synthetic oligonucleotides. They use the AutoFlex Speed MALDI system to quality control their oligonucleotides. The overall throughput is typically 1,000 to 2,000 samples each day. And both MALDI and electrospray ionization are used. The first pass is MALDI and the second pass is the QTOF for samples that provided poor quality MALDI spectra. This slide shows the results on a MALDI plate that was used for screening oligonucleotides. The results are seen in a traffic light reporting based on the multi-attribute criteria that was set by the user. All of the attributes set show a green code in the table, indicating that this particular sample passed the multiple criteria set by the user. The expected table in the results view shows in greater detail the modifications, while in the spectrum in the lower half of the figure, the molecular ion and the modifications as well as different side products that are present are annotated. The last set of examples come from Kaneka Eurogentec. Eurogentec is a 30-year leading supplier of high-quality, custom-synthesized oligonucleotides. They utilize the BioPharma Compass software for routine oligonucleotide QC that can be applied to both electrospray ionization and MALDI data. The advantage of the MALDI platform is the increased throughput, while electrospray ionization, with its higher M to Z range, provides superior data for larger oligonucleotides. For MALDI analysis, the AutoSec speed is used. Oligonucleotides are prepared in a 3-HPA diammonium citrate matrix using robotics to spot a 384 anchor chip sample plate and then analyzed in the MALDI-TOF in linear positive ion mode. On the electrospray side, the compact QTOF is used. The samples are first desalted on line using a C18 pre-column. DIEA is used as an ion pairing agent, and they're then eluted using acetonitrile and detected in the QTOF, operated in negative ion mode. This slide shows an example of a small spectrum confirming an 18-mer. Here you can see the molecular mass at about 5,000 Daltons and not too many modifications. All of the criteria have a green color coding, meaning that the sample passed the QC criteria set by the user. Next, we see a larger sample. This is a MALDI spectrum of a 34-mer. We can see the mass at about 10,000 Daltons. Not too many modifications. And again, all of the criteria have a green color coding, indicating that this sample passed the multi-attribute monitoring as well. The third sample is for a larger oligonucleotide. This is a 55-mer, and as you can see, the mass is seen at around 17,000 Daltons. In the table, if you notice, there are some attributes that have not passed. So we see a red color coding for the base peak and a yellow color coding for the intensity coverage. So this sample probably needs to be run again or checked by another method, like maybe ESI. This slide illustrates the superior data that can be acquired for a larger oligonucleotide with electrospray ionization. This is a 65-mer, and the oligonucleotide peak can be seen at 19,000 Daltons, while the modifications are clearly visible as well. Electrospray ionization gives a detailed characterization of the oligonucleotides. The average spectrum seen in the central window and the deconvoluted spectrum seen in the left window both indicate the increasing background ions and side products. These are also seen reflected in the multi-attribute table, where the scores are reduced, as seen by the green colors changing to yellow and red for failed attributes. In conclusion, for oligonucleotides, a complete solution is presented with a range of MALDI and electrospray instrumentation and a wizard-based software platform, BioPharma Compass, to analyze the data. Please see the poster that will be presented at ASMS 2019 in Atlanta, as well as the flash note on our website for further information. Please do not hesitate to contact me at the email address below or for any other information that you would like. Again, thank you for your attention, and I would be happy to answer your questions.
Great. Thank you so much, Anjali, for your presentation, and thanks everybody who was listening to the presentation and sending in questions. Just a reminder again, you can submit questions right now, and we will try to get to them in this following Q&A session. So Anjali, I wanted to start with some of the questions that we have here that have come in. Okay, so one of the questions is it possible to differentiate deletion of an A versus a G?
Yes, it is possible to differentiate those. There is a difference of 16 Daltons between the A and G, so you should be able to see that difference on the spectrum.
Another question that came in asks, how long does it take to prep a 96-spot MALDI plate?
So this would depend on how the prep is actually done. If one does a manual preparation, it takes about a couple of minutes per spot. And if you use an automated spotting instrument, then this would probably take about a few minutes for each plate. Again, that depends on the actual instrument that's used for the automated spotting.
Another question asks, I'm wondering if ESI data were acquired with HPLC separation. How about MALDI with HPLC?
Yes, the ESI data was acquired for using HPLC separation, and the MALDI data was not done in this instant with LC separation. It was just spotted directly on the plates. It is possible if you have an LC that you can connect, then you can do spotting of the separated fractions as well.
Can the spotting of samples on MALDI targets be automated?
Yes, it is possible to automate the spotting on a MALDI target. For example, one can also use the RapidFlex MALDI Pulse high throughput system, and this is specifically designed for fully automatic handling of 384 and higher spot plates. This also includes automated method development and export to a LIMS system.
How can you tell if depurination occurred in the mass spec source or it was a real impurity?
Sorry, I didn't get that one word that you mentioned. How can you tell if depurination?
Depurination occurred in the mass spec source or if it was a real impurity?
So that is a bit difficult to say. I mean, of course, a lot of it depends on your source. Typically, the electrospray QTOF sources that we have are very gentle, and so we don't really see a lot of depurination and other modifications in source. With the MALDI, again, that's another question because sometimes it is possible to see such happening on the source itself.
Have you done any sequence analyses, either enzymatic ladder sequencing with MALDI or MS sequencing using electrospray?
Yes, we have done sequencing analysis both with the MALDI and with MS as well. On the electrospray side, sorry.
Yeah. Could you estimate purity of oligonucleotides from HPLC ESI MS or MALDI MS data? On the electrospray systems, yes, you can estimate the purity using the base peak information that you would get, and that's relative purity. So you would be able to estimate that. On the MALDI side, I'm not too sure about that one. I think you would get a better estimate from the electrospray information. Is there a preferred matrix for the high throughput MALDI TOF applications, or is it dependent on the size of the oligonucleotide under investigation?
The preferred matrix is HPA, which is the 3-hydroxypicolinic acid for the MALDI.
How long does it take for a novice user to learn how to use the oligonucleotide part of BPC?
So since BioPharma Compass has a specific oligo workflow, it has all the necessary parameters which are required for the data processing of an oligonucleotide. And so it really doesn't take that long for a new user to go through the workflow and adjust any parameters depending on their experiments as required for the processing. So it should be fairly easy.
For GLP labs, what tools are available to prevent tampering with results or raw data?
So there is an audit trail which is included in the regulatory toolkit, and this is optional. It's a built-in audit trail, and all changes are tracked on the server's audit trail. There is also mandatory justification if configured. It is also possible to get audit trail reports which list all the user interactions that have led to the particular result.
Is the software specific for oligonucleotides, or can it also be used for peptides and proteins?
The software has workflows for both oligonucleotides as well as proteins and peptides. So you can choose the specific workflow of interest, and the parameters will be adjusted according to whether you are looking at an oligonucleotide, an intact protein screening, for example, or on the peptide side, whether you're looking at peptide mapping experiments. So all of those are built in and easy to change.
Another question that came in asked, were the spectra signals shown deconvoluted? Is maximum entropy deconvolution feasible for oligonucleotide?
Yes, the spectra signals were all deconvoluted, and maximum entropy was used for the deconvolution.
Another question asks, can MS data acquired from other softwares be analyzed with BioPharma software?
So I'm not sure about this one. I'll probably have to get back to you on that. So this is specifically for the oligos. I'll have to look into it. I know that for the proteins and other workflows, it is possible to look at data acquired from other instruments. So my guess would be that it should be possible for the oligonucleotides as well, but I would have to get back to you offline on that one.
Another question that came in asked, are there recommended calibration standards for your MALDI TOF instrument?
Yes, there are recommended standards, and we do offer a calibration kit for the MALDI TOF instruments. But having said that, it's also possible to use a lot of other more readily available standards that you can calibrate the instrument with.
Another question that came in asked, can one of the shown instruments differentiate between PS-PO exchange and an AG exchange?
Sorry, what was that question again?
Oh, that's okay.
Don't see that one.
So this one asked, can one of the shown instruments differentiate between a PS-PO exchange and an AG exchange?
Yes, I believe there was a slide. I think this was on the electrospray instruments, the compact, and it is possible to differentiate between the PS-PO exchange and also the AG exchange.
Another question that came in asks, are there any special sample preparations necessary to ensure that an oligonucleotide sample has stability?
This would really depend on the sample prep for whether it is a MALDI or an electrospray instrument that we're looking at. But as far as stability goes, it would really depend on what instrument, what platform you would be using.
Does the software automatically look for N1, N2, etc. oligos?
So the software will look for the specified modification and annotate this as well. This would be seen in the expected tab. Other peaks that are present, but they are not, which were not part of the method, are also shown in the unexpected tab, and then you can look at those for further review. So you will see both the modifications that you have specified as well as ones that were not specified.
How do you handle batches?
So the automated acquisition tab, which is part of the screen workflow, allows you to queue batches for acquisition. Batches can be prioritized as well. And you can import sample information via a CSV file, or this information can be manually entered as well. So all of that goes into the batch. The batches are run together, and the processing tab will tell you when a batch has been completed.
Another question asks, how is the software licensed?
So the BioPharma Compass license is based on the number of concurrent users. The floating license system makes it affordable to install the software for processing or reviewing the data on large number of computers. And the system can also be scaled as needed by adding licenses to fit the number of users that are logging in at a peak utilization time.
What is the largest molecular weight that can be measured with isotopic resolution?
So this again would depend on the instrument and to a certain extent on the application. On the electrospray side, the Maxis instrument has the highest resolution, and you can expect an isotopic resolution between 55 to 60 kilodaltons. The compact is a lower resolving instrument, and with that, I've been able to see something around myoglobin size, so about 17 kilodaltons isotopically resolved. On the MALDI side, this is again very dependent on the matrix used, and so the RapiFlex has the highest resolving power, while the AutoFlex has a lower resolving power. But it really depends on the sample prep and what matrix you're using.
Okay, great. That's all the time we have. Thank you again, Anjali, for your fascinating presentation. And thank you, participants, for being a great audience. Be sure to check CNN or CNN Online for information on the next edition of CNN webinars. Thank you, On24, for technology and production services. And thank you, Bruker, for the sponsorship that made this interactive webcast possible. For CNN webinars, I'm Jeff Huber. Have a great day.